Hello, and thank you for taking the time to watch our video and to learn more about Vivet Therapeutics and our clinical trial for Wilson disease known as Gateway. My name is Lori McKenna, and I work in patient advocacy for Vivet. I'm based in Boston, and I am part of the expanding team at Vivet. And with me is my colleague, Dr. Bernard Binichou. He is our Chief Medical Officer at Vivet, and he has been with our company since its inception nearly 7 years ago. He will share some background on gene therapy and about our own clinical trial Gateway, and hopefully start answering questions that are on your mind along the way. So here's a quick look at our agenda. We'll provide an introduction to our company, a little bit about gene therapy and about our investigational therapy known as VTX-801. And we'll talk about the meaningfulness of gene therapy specifically for Wilson disease, and of course about our clinical trial. So here's a quick look at our team on the left. We are based in France, and we are a clinical stage biotechnology company. The people you see there are entirely devoted to making a difference in the lives of those with rare and inherited metabolic disorders, with our lead program being for Wilson disease. And at this point, I'd like to turn the presentation over to Bernard.
Thank you for this introduction, Laurie. So about 30 years ago, I attended one of the first congresses of gene therapy. And at that time, we were wondering, is it going to work? Is it going to be efficacious? Is it going to be safe? What will be the long-term effect? I'm happy to say now, as I said, about 30 years later, that many products have been approved for treatment of patients, for treating rare disorders, inherited disorders, or for cancers, and targeting the brain, the muscle, the skin, blood, the eye. So about 19 products have been approved so far, and I'm pretty sure that in the coming years, there will be more products. As you can see, 2 products have been approved for hemophilia. That's of interest for us because they use a technology that is very similar to what we are using for Wilson disease, and they also targeted the liver. In this case, for hemophilia, they use the liver as a factory to produce the missing coagulation factor, and this was successful. Next slide. I also want to share with you some very recent results that have been published in the New England Journal of Medicine. So on liver disorder, it's called Crigler-Najjar, very rare, but it affects the liver, and these patients need a treatment for life to survive. And patients have been treated now with gene therapy a few years ago, and this has been successful, and they were able to stop their standard of care treatment. The product has not been approved yet, but this is extremely encouraging for the future. And again, like for hemophilia, they use a technology that is very similar to what we are using for Wilson disease. Next slide. So as you know, in Wilson disease, the defect that is associated with the disorder is a defect in a copper transporter called ATP7B. And this ATP7B copper transporter normally acts in the liver to eliminate the excess copper. If this ATP7B copper transporter is not functioning properly, then copper accumulates in the liver and in the other organs, especially in the brain, and this leads to the symptoms that you know about the disorder. So the objective of gene therapy is to restore this function of copper transporter to allow for a proper elimination and regular elimination of excess copper from the body. Next slide. For that, we use what we call a vector. A vector is actually the empty shell of a virus, just the shell of it. We don't take the viral DNA so that this is not infectious. And in this shell that we use as a shuttle, we introduce the gene of ATP7B, the corrective gene, and we use the whole thing to this vector. We inject it intravenously in the patients. The vector is intended then to circulate into the bloodstream to reach the liver cells, and then to modify the liver cells so that they will then be able to restore this function of ATP7B that can eliminate the excess copper from the blood. If you want to learn more about gene therapy for Wilson disease, I invite you to scan the QR code on the right-hand side of the slide. This is a very nice, there is a very nice video produced by the American Society of Cell and Gene Therapy that really explains what it is. Next slide. So this whole program of VTX01, that's the name of the product that we're using. This whole program is based upon previous experiments, as you can imagine, especially experiments in animals and in mice, where we demonstrated the activity and the efficacy of the product. We use a slightly different version of the shell so that it can really work in mice, but otherwise the core, let's say the DNA piece is identical to what we're using in humans. And what we did is the following: we used a very small amount of radioactive copper that is not dangerous to the mouse, and we injected it into the blood of the mice. So what you see on the screen is some kind of an X-ray. You see the whole mouse, and what you can visualize is the radioactive copper as this light blue signal in the liver. Naturally, copper will go into the liver and then will be eliminated below, as you can see, in the intestine. This is this blue signal, and this is the normal pattern that we should observe. Now, if we look to the second mouse, which is untreated, after the injection of radiocopper, we see that there is a strong signal in the liver. It's in red, and there is no signal in this testing showing that copper has been trapped into the liver. This is absolutely abnormal, and what we want to restore is the picture on the left. So what we did next is that we treated, and on this picture we show 4 different mice with Wilson disease, and we treated them with increasing amounts of VTX01. And what you see is that progressively, with increasing doses of VTX01, then the signal in the liver is less and less intense, meaning there is less and less copper that stays in the liver and more copper that goes into the intestine. And with the highest dose, actually, we observed a complete restoration of the normal pattern that we were wanting to observe. Next slide. I'm not going to show you all the experiments, of course, that we performed, but another one is very important. We looked at survival. And so what we did, simply, we treated some mice with VTX01, and others were not treated, and we observed them. And after 44 weeks of age, we started to see the untreated mice dying, and the other ones were not in a good shape. By 1 year, 60% of them were dead. And on the other hand, those mice that had been treated with VTX01 were surviving and were perfectly healthy. Next slide. So now, what is the ultimate goal of gene therapy? We want to restore the elimination of excess copper from the body because it's toxic. That's what we just discussed. And this should allow us eventually to restore the standard of care treatment with a scalar or zinc treatment, and also to interrupt the low copper diet. We want to prevent the disease complication and hopefully improve existing symptoms, including neurological symptoms. At the end of the day, what we want, the ultimate objective, is to improve the quality, the life of those patients, of those people living with Wilson disease. On the other hand, what gene therapy is not expected to do is to restore the function of dead neurons. If neurons are, tissues and neurons are dead, then it's unlikely that gene therapy will improve that. So this means that in the future, we'll need also to intervene as early as possible. Next slide. So let me update you briefly on where we are with Gateway. You know, this is an ongoing clinical trial. This is the first one with VTX01. And the objective of the trial are first, as is always the case, to ensure to evaluate the safety of the products. And also we want to collect some preliminary efficacy of a single administration of VTX01. The second important objective is to determine what will be the optimal dose to use in a larger trial. Let me now briefly talk about the visits. During the first 3 months, there will be what we call an intense monitoring of the patient, so frequent visits to ensure both safety, but also to maximize the chances of success of the therapy. We feel this is very important. After 3 months post-injection of VTX-01, we should be able to determine whether we are able to determine whether the patients are responsive to the treatment, responders, yes or no. And the patient, if a given patient is a responder, then we withdraw the standard of care treatments and we continue the observation of the patients. If the patient is not a responder, then we let the patient under their standard of care treatments. During the afterwards, we will progressively decrease the number of visits. During year 2, there will be 4 visits, and then during year 3, 4, and 5, there will be 2 visits per year, which is comparable to standard of care management. So far in the trial, there has been no serious adverse event that has been reported after injection of VTX-01, and the enrollment continues. Next slide. So this is very briefly the trial. There will be about 16 patients that should be enrolled at the end. The key enrollment criteria, there are many more, of course, but the patient needs to have the Wilson, to be an adult between 18 and 65 years of age. And the patient needs to have been treated on the standard of care for at least a year. And the total duration, as I mentioned, is 5 years. Next slide. Initially, we had 6 sites across the US and Europe, and we have expanded that to 10 clinical sites to make it easier for the patients so that they're closer to the sites, and it's certainly much more convenient now for the visits. The lead investigator is Professor Michael Shiskey from Yale in New Haven. In the US, we have another 5 sites with Fred Ascarin in Michigan, Valentina Medici in California, Regino Gonzalez in Florida, Will Lee in Southwestern in Dallas, and Sean Rudnick in North Carolina. In Europe, we have 5 sites: Thomas Sandal in Denmark, Ulrich Lauer and Hartmut Schmidt in Germany, and Aftab Allah in London in the UK. Next slide.
So thank you very much, Bernard. We're almost at the end of our presentation. This is our last slide. Please know how much we appreciate what an important and personal decision it is to participate in any clinical trial. And as Bernard said, we are doing our best to take away some of the stress that comes with that decision. We have added more clinical trial sites, and we have a team devoted to answering your questions, your very personal and specific questions along your journey. And for those who are eligible, there is assistance with travel and study-related expenses available. So I encourage you to scan the QR code on the left of the screen and to learn more about our study. So thank you very much, especially to the Wilson Disease Association for providing us with this opportunity today. And thank you for taking the time with us today. Thanks so much.